Okay, so just as our panelist is getting seated, I'm just going to introduce them. We're just going to give their name and where they're working, and then they're going to talk a little bit about what they do on the panel. So here at the head of the panel here is Sonia. She is our oilseed specialist. We're very glad to have her on board with Manitoba Agriculture now. We have Chris Manchur, who's with Canola Council, and then we have Meghan Rose, who's with New Era out of Swan River, and we have Evan Gillis, who's with D&L Seeds.
Okay, so we're going to start. We just wanted to do, from different perspectives, just talk about the disease situation in canola. We have some slides we might reference, but really just kind of try and do a miracle and cover verticillium stripe, blackleg, and hopefully sclerotinia here. So our first question for our panelists, we're just going to ask, what kind of is your role and how are you working with canola and with disease kind of in your day-to-day? So start with Chris.
Yeah, so this is kind of a tall order, I think, for our panel here because it's a lot of different topics to cover, but I think we'll do our best. Anyway, so yeah, I'm with the Canola Council of Canada. My official title is an agronomy specialist, so we're a team of about 8 or 9 of us across the prairies. And not only do we have territory responsibilities, providing agronomic support, extension services, we also have specializations, which includes looking at, whether it's breeding, pathology, entomology, all those kinds of things. So my responsibilities is for the eastern half of Manitoba, as well as sclerotinia leadership, which also includes, working at the sclerotinia steering committee. I know Kelly was on there. I don't know where he is right now, but and then also with Clint Yerke moving on to BASF, I'm also now taking the lead for blackleg as well. So kind of double dipping on some of those responsibilities there. But yeah, I'll pass it on.
My name is Meghan Rose. I'm from Swan River. I'm the research manager for New Era Ag Research. It's a company associated with an ag retail in the area, and so my role is just basically a contract research manager, and so we do a lot of different trials with crop inputs for canola, disease management, as well as working with breeders, screening some of their lines that are hopefully going to offer some protection against some of these diseases that we're seeing in the field. So.
All right, Evan Gillis, canola breeder at DL Seeds, so I'm part of a team there that is working on developing varieties. Little known company, we don't have a full marketing wing. We're not selling direct to farmers like some other companies would be, so we are kind of a purely research and development, variety development company in canola and dabbling a little bit in pulses. So you would see our varieties with like a Brett Young or Cantera type of a marketing partner. And so we're a little bit in the background, but hoping to bring some insight from what a developer is going through trying to bring products to market that have disease resistance.
Okay, so we're going to tackle the elephant in the room first, and we're going to talk about verticillium stripe this season. So maybe I'll head this question over to Meghan first. I guess kind of what did you see over the season, in terms of verticillium stripe, and how were you finding it in the field? And I'll put up some pictures as well here.
I think it's probably surprised everybody a little bit, the amount that we were seeing. Compared to previous years, and we had so much sclerotinia this year that I think even when farmers were seeing it, they were just saying, "Oh, it's sclerotinia." It does look quite a bit different to the trained eye, but sorry, what was the question? How do I?
Yeah, well, I guess just what were you seeing, and what are some things like to identify it? What are you looking for?
Okay, so I guess we're seeing a little bit later than what we would see the initial sclerotinia symptoms, and so it's kind of past that flowering stage, but the first thing that you see is that striping, which I recently learned doesn't always necessarily mean that you're going to have a lab confirmed case of verticillium, but that is the first thing that we see in the field. Sometimes that turns into a percentage of the plant that just doesn't have the vascular tissue to fill those pods properly, and so they dry up, premature death, just a lot of brown stems where you would be expecting sort of a greener, still lusher crop. There is the cross sections also that you can do at the base of the stem, but that's a little bit more tricky to distinguish between blackleg. And then later in the season is when you start to see the shredding of the outer layer of the epidermal layer of the stem, and there's the microsclerotia in there, which also sort of look a little bit like blacklegs. So if you're not sure of the difference, it can be a little tricky to ID.
Yeah, so maybe we'll go into that a bit. If you are looking at verticillium versus blackleg in the field, and I'll open this up, what are you looking for, I guess? I have a slide here somewhere, but.
While you're looking for that,
Yeah.
I think just something to consider too is what you're looking for really depends on when you're actually scouting in those fields, because we have different times when we want to get out into those fields. For sclerotinia, I find it really nice to go in a little bit earlier because you're starting to see that initial affection that's happening maybe higher up in the canopy. But also we can go and take a look at blackleg and verticillium, at post-harvest or, at swathing timing and such. And the symptoms really do differ. Even when you get kind of later into the season too, you're starting to get some of the necrotrophic kind of pathogens that's feeding on decaying tissue as well. So that makes things even more confusing. But looking early
and
on,
the fact that all three of those things are sometimes in one plant, so they're confounding each other.
Totally, yeah. I think early on, it's probably the trickiest for verticillium stripe because you do see kind of that cross-section discoloration. I see some of the images we have up here. On the right picture, we have a pretty mature kind of stubble sample that shows verticillium stripe. And I remember I did take a picture, and I should have sent it to you, but it just starts to begin this kind of like gray-brownish starburst pattern starting from inside because it's starting to infect all that all that water and mineral transport vessels coming in, and that's kind of the first step. And then it starts to decay more in general rather than blackleg that gets that kind of wedge-shaped patterns, but unless it's severe, it gets pretty overcome.
Well, I'm gonna move to Evan here. So how are we seeing verticillium affect canola? We mentioned a bit here, the vascular system, etc. But from like a developer's point of view for varieties, what are you seeing?
Yeah, I think Chris was just about there. I was just going to jump in with that comment. One thing just on that comment and on the picture on the screen, it's causing problems not only on its own, but for a developer trying to develop blackleg resistance, for example, that's really messing with our nurseries and our ability to just test for blackleg. So this combination of multiple diseases in the plant, it's really hard to identify what's going on with either one completely. So it's sort of causing some confusion and causing some problems for the industry to really properly assess blackleg as its own disease. And also, obviously, there's a bit of a connection there. Potentially, verticillium blackleg is a bit of a complex going on of some sort that needs to be dealt with.
Yeah. So, as this is kind of a, I guess, newer topic, we're seeing it more potentially. What are some of the things that you would want to see, just from a canola perspective, some of the tools potentially to kind of combat verticillium for the industry? I'll open it to, yeah.
I think our most mature disease that I would work with is blackleg. And so we've got, long-standing steering committees. There's been multiple sort of larger consortiums with academics, government, industry companies. All of that stuff, that path to being successful against a disease, I think is on the go right now in verticillium, just it's sort of in its infancy. So all of that stuff needs to kind of make its way in for the industry to be able to have the tools it needs to fight the disease. But so that's all well and good, but right now it doesn't appear that we have a blackleg-like situation going on where you have a gene-for-gene interaction. As a developer, like what I always want in our breeding program is a single gene and a marker that we can just cross into our plants, and it doesn't appear that a disease like verticillium is quite there yet with a bunch of genes that are available to the industry.
Verticillium breeding probably is more akin to, say, sclerotinia breeding for resistance in terms of all these little minor factors in terms of for selection and such. And both sclerotinia and verticillium are generalist pathogens that affect many other crop species. So while the situation would be fairly relatively new for canola, It's been with other species for a while. You know, in potato, it's the potato early dying disease, and so we kind of see those parallels with canola, where all of a sudden the canola starts to die off maybe a little sooner than we think, and then that's lopping off that upper yield potential for us. So yeah, genetics would be the golden goose for that, but the unfortunate thing is we don't really have many other chemical or cultural control methods that are like really put this thing to bed. Similar to like clubroot, where you know a 2-year gap is very effective in managing it. The research isn't maybe quite there in terms of verticillium.
Yeah, and I think that's what we maybe need to focus on in the field is trying to find some cultural practice or something that we can do to at least slow it down or mitigate it, and because it is going to take some time before we see anything come through in the varieties.
Okay, any last thoughts on verticillium? Or we might move on to blackleg.
I don't want to be too doom and gloom on things, but I'll hit it from both sides here, a little bit of a positive and a negative, but just on the flip side, just something to think about. We've been working with our parent companies in Germany for a lot of years, and this disease has been around there for a long time. There's experts in Europe that have been working on it for a lot longer than people have here. And it was always kind of thought of as almost like a harvest aid when straight cutting canola there. It helps with dry down. I know some sort of complaints or some comments I've heard around the industry about really green stems going through the combine and how some new varieties, that's an issue. So I'm not saying we're going to try not to breed for disease resistance, but something to think about is there a balance? If you have too green of stems and your plants aren't drying down, is there some benefit to a little bit of late season disease? I know there's pockets like Portage area that we've seen devastating verticillium outbreaks, and we clearly can't have that, but just there's maybe not, it's not all doom and gloom is what I'm trying to say, I guess.
Yeah, and a quick shout out to even the U of M who, you know, talking about European experience, we're importing that into Canada with Harmi Chawla coming from those labs and research institutions out in Europe, and being part of some research projects that, you know, growers such as yourself as a director on the Manitoba canola growers are funding to better understand this disease. So that way we can actually hit the ground running and help accelerate some of those breeding efforts.
Okay, so with that, I think we're gonna switch into blackleg here. So I'm gonna field this question first to Chris. So how are you seeing blackleg in the field? What is it? Maybe give us a bit of background here.
So with the fall, it's really heavy getting into extension season and then getting all the reports from all the 3 prairie provinces. And so I've probably heard the pathology reports from the provinces probably about 4 or 5 times now at different instances. And blackleg is probably our #1 pathogen right now across many areas in terms of yield losses, productivity losses. I know Manitoba, we have verticillium very big. There's some clubroot in Alberta. But in terms of a whole prairie-wide approach, blackleg is definitely significant. And so it's definitely something that I think we need to make sure we're keeping on top of, staying on our radar, and I'm sure we're going to be talking more about the management. But in terms of prevalence incidence, it's just keeping going up. And so if we're taking a look at prevalence, I think it's found in almost every field across many of our provinces. Incidence is slowly rising as well. When you're starting to take a look in the late 2000s and in the 20 teens, it was fairly low, and now later 20 teens up until now, it's just been starting to steadily climb.
Yeah, and I think one thing that surprised me this year was just being in the field and seeing different localized hail events, and then seeing the blackleg infection on the stem as well this year. Saw that a bit more as well, just in. I guess for Meghan here, how do you see blackleg affect canola yields? Are you seeing it a lot kind of in your region up north as well? Yeah.
I think since farmers switched over to straight cut varieties, there's a lot less scouting for blackleg at the correct timing, so it's maybe not being identified as much as it should be in the fields. I don't really think it's on anybody's radar in Swan River because we had so much sclerotinia and so much verticillium, and they're much more visual than blackleg if you're just, well, from the combine, I guess. So, but as far as how it affects the canola crop, it's just restriction of flow of water and nutrients and poor pod set and smaller seed and reduced seed yield. yeah. But I don't know if there's a link between the transition to straight cut and our increase in blackleg, if that's a link at all, or that's what I see.
Well, and so for Evan, so what in terms of, I guess, kind of genetics, just from the developer's point of view, you know, blackleg, I guess, has been around for a while and there's some options, but kind of what's kind of your view on that?
Yeah, I think I touched on that a bit in what my sort of dream for verticillium would be to be at right now. So yeah, blackleg is a much more mature disease as far as what the industry has been doing with it. I think we're pretty well set up with markers and different genes. I don't know what we're working with, but it would be more than 10 different genes we'd have access to that are published and ready to go. So it's a matter of what's most prevalent out there and how quick we can adapt to that. And of course, everybody will always preach tight rotations, make it difficult for breeders to keep up with those things. So certain areas with every second year canola or worse, we do see some wrecks in fields due to blackleg, and it takes something like 3 to 4 to 5 years, right, to get a new gene to the market, even if it's sort of already on deck. So that is just the classic warning of it's a little difficult to keep up with some of that stuff in the breeding side of it. Just takes time.
So are there any other, I'll just open this up for everyone, any kind of management considerations, I guess, around blackleg? We've touched on a couple, but other than genetics, that you'd consider?
It is very much a layered approach, in my opinion, to managing blackleg. We've talked about genetics a lot, and genetics are very effective when deployed properly. And when you're deploying genetics, you really need to understand what you're working with if you're in a high-risk situation, and then also understanding what kind of genetic packages you have. You know, we have qualitative resistance as well as quantitative resistance, and both can work, but you have to understand the situations where you want to apply that. And then when you pair those genetics and your risk situations, you know, relating about rotation, then maybe you need to also consider, you know, applying that blackleg seed treatment if available, or if you need to, if kind of a worst case scenario, if you really also need to apply a blackleg fungicide. And blackleg fungicides can be effective if they're timed very well, and usually they're very economical because you can put it in with, say, your first pass herbicide mix, or maybe with your flea beetle insecticide. But when you're taking a look at, a wholesome approach in terms of that, there's all these different things that you can consider for this. But really it all kind of ties back to understanding what kind of situation you're being dealt with, which means scouting the field. Some people probably don't know how bad blackleg is. You know, blackleg severity is rated on a 0 to 5 scale, and you don't have to have a 5 rating to say, oh, we have a problem. You know, 3 out of 5, you can start incurring some significant yield losses. Yeah, exactly. So like I wouldn't want to see anything, you know, above a 2 before I really need to start considering, okay, I need to change my practices a bit.
Yeah, and sometimes you won't, right, necessarily see on the stem the blackleg infection, but when you cut the cross section, you know, seeing it. So that's even another example of just getting into the field. I really found that during the disease survey as well this year. Any other comments, I guess, on management, etc.?
I guess blackleg is maybe a little bit, the good part of it is a residue-borne disease, and so once that residue breaks down, it's going to disappear out of the field, whereas verticillium is, those microsclerotia are going to persist in the soil for a lot longer. So rotations will help with blackleg.
I think the monitoring side as well to help the breeding side. So there's what's going on with the different pathotypes in the field. What are the targets? What are the major lookouts? So there's been some of that work that's gone on over time, and it's easy to target certain genes. Chris touched on something that's really important. We do a bunch of screening. We have several disease nurseries. The quantitative side of it, I've talked about major genes, but the quantitative side of it's really important as well to have that background. And it's just there in a line, and you have a lot of protection from that. So that's the foundation of the breeding program, and then the genes are added on to target certain regions or whatever after that.
Okay, awesome. So, we've covered 2 out of 3 so far. We're doing well in terms of time. We're gonna move to kind of our final last disease here and then open up for some questions. So we're gonna head into sclerotinia. So, Meghan, as you mentioned, Sclerotinia was around this year. So I guess what were you seeing in the field? And I'll put up a really good picture from Evan here as well.
We definitely saw it everywhere, every field, and I guess every field that I went into, but I was also targeting, I was looking at bad fields. So if I tried to find the best field in the area, I could probably still find some. Colleagues of mine have said it was the worst infection rate in 25 years and caused more damage, more yield loss than he had ever seen. So that's a lot longer than I've been around to be paying attention to Sclerotinia. But yeah, it was, even in the fungicides worked, but it wasn't 100% cure. It didn't, it wasn't completely clean. And where we did have fungicide, then the verticillium really showed its face.
Yeah. Did Evan or Chris, did you guys see Sclerotinia around this year?
Well, that picture up there is a field that I went through. So that field ended up with a lot of disease at the end of the day, but this is a sort of sprayer tank run out / check strip situation that a friend of mine had out in the field. And this is pretty early on, sort of right at the end of flowering, or a little bit past that maybe, but just a devastating amount of Sclerotinia, and that's basically all that was out there. So that was more or less a total loss. And I guess my comment on Sclerotinia, and I think Chris touched on this, is it's tough to work with. It doesn't have, there's no like major gene like a blackleg like we talked about, so it's a tough one to work with. There's a lot of, the fungicides are effective. I think this year it rained a lot and for a long period of time, and the flowering window is long, so with one pass of fungicide, it's probably not enough to cover that whole time, and if it's really high risk, you're gonna be in trouble. But because the fungicides are so effective, it's a lower priority compared to a verticillium or something like that for a breeding company. As a general comment, it's difficult to work with, and there's effective options out there. So that is an unfortunate thing with this disease as far as genetic improvements go on it.
Yeah, and so maybe I'll head this to Chris. So what are some of the, I guess, kind of management options, and what are they helping with in terms of, you mentioned fungicides, et cetera, but what are they helping with in the canola, like the fungicide, et cetera?
Right, yeah, so Cole's note summary for fungicides, you want to apply your fungicide to protect the petals of the canola plant, right? The more petals you protect, because that is the primary mode of infection once those ascospores are released from the apothecia coming from the soil. And so that's where fungicide timing has a really major role to play for that. And I've seen some cases where, you may be still applying a fungicide, but if the timing is off, you're still seeing significant levels of infection. And in high-risk areas where we saw like fungicide, yeah, it's there, but you still have unprotected petals or maybe a later season infection, or if you sprayed late, the infection came earlier, right? So that's one of the chief tools, and yes, it is effective when used correctly. The other factors into that, and this kind of folds into kind of the integrated pest management strategies, what are kind of environmental factors that you can potentially manipulate? And when I'm talking about manipulating the environment, and I'm saying stop raining, in the last 2 weeks of June, I'm talking about, influencing the microclimate for your plants. And so if you have a really dense canopy, a really dense stand, and one of my colleagues, Keith Gobbard, all of his presentations, he goes and brings a huge wet pants showing about that kind of simple test that if that climate is really humid, really wet, that's excellent conditions for that. So if you're being able to reduce your plant stand, or maybe conversely, understand that your risk is going to be elevated for that, then that helps. Now, talking a little bit, and Evan touched on it, it's really difficult to breed. There is always going to be some sort of selection bias in terms of, advancing Sclerotinia resistant cultivars, and maybe resistant isn't the right term, maybe a little more tolerant. And so there is going to be a subtle progression through, and there's always plenty of research taking a look at Sclerotinia resistance and, genetic lines, germplasm that has varying levels of efficacy, but it's really difficult to get it into a high-performing hybrid, I think. And I don't know, maybe Evan, if you confirm that, or you think, oh yeah, it could be easy.
Yeah, certainly there is people in the industry that have like pretty solid hybrids on Sclerotinia out there. It's a lot of work, and it's difficult, yeah, compared to some other diseases for sure.
So, yeah, I think that's, really interesting. In terms of, I guess, kind of sclerotinia management, you know, Meghan, like how very practically are you approaching in a high-risk year, just kind of scouting your field, et cetera? Like what are you very practically doing?
In our area, it's very common to spray. Everybody has that penciled into their canola crop cost. I think the general feeling is if the crop looks good and it looks like there's going to be some bushels there, you spray it to protect it because you don't know what's coming. If you have a thinner stand, the microclimate within that is maybe a little bit drier. If the season has been drier, then or that doesn't look like it's going to yield, then maybe that's taken out. But for myself on the research farm, it's just practice that we spray, and we're doing that at 30 to 50% flour. It was interesting this year, we did have kind of a long canola seeding spring, so some stuff was in really early and some stuff was in really late, and so when we had the rain events and the storms that really pushed that crop down, things that were already flowering, crops that were already flowering were much harder hit with sclerotinia than the crops that were, you know, still bolting, and they kind of withheld that windstorm, and so that could be a management strategy, I guess, to spread out your risk. Don't seed it all as soon as you can. When I first started, I guess, we were coming off some wet years, and it was general practice on the research farm to spray twice, and then we kind of dropped that in the drier years. So maybe we're heading back into that, I'm not sure.
Okay, I'm going to ask more of an open-ended question, then we'll go into the audience here if they have any. I guess what is something, you know, we've covered blackleg, we've covered verticillium, sclerotinia. What's something that you would be top priority for you and you'd want to see in terms of kind of canola disease protection? And what is also something that has been a success, I guess, as well? So, bigger open-ended question, and I'll open for anyone here.
Okay, I can. So, what's been a success and what would we want?
Yeah.
Okay. So, I mean, I think most of, I keep going back to blackleg because it's easy, and it's relatively easy, but that's been somewhat of a success. There's a lot of published genes available and all that. We haven't touched on clubroot at all here today, but that was a very scary situation in Alberta, and it's here in Manitoba as well. And but I think the industry responded pretty quickly to that with some quite effective genes that are out there. So I think that's quite a success as well. I think what I look for is, we're talking about, you know, verticillium here now in a reactionary way, where it's become a major problem in Manitoba. I've talked to farmers around Portage that are cutting their acres. They're considering getting canola out of the rotation for other crops. Pretty major issue in a relatively short period of time. So this sort of reactionary approach to things is something would be nice to see a longer term thought process. So there's things like turnip yellow virus or leaf spots and things like that in Europe that are likely to be here at some point, possibly. We had that discussion about clubroot. It's just a Europe problem. Now it's not. So I think that sort of long-term vision, which is really difficult to find budget for, obviously, is one of the things that would be nice to have a little more of.
So before I kind of declare one of our bigger successes, I just want to kind of poll the crowd by a show of hands. Who here has been able to identify verticillium in any canola field?
Ask the opposite question.
The opposite question would be a lot more difficult, but I see a lot of hands, and so I think that's one of the major successes in recent time that we've seen right now, is that we've taken big strides, you know, just in the past 3 years of our understanding and be able to identify verticillium stripe, because in order to really develop a good threat assessment and then also direct resources, whether it's breeding efforts or, you know, other alternative modes of control or management, we really need to understand what we're dealing with. And so to me, it's always the identification of the problem and the severity of that problem. And so it's nice to see a lot of people can go out in the field and identify verticillium. We're going to be, I know the verticillium stripe steering committee is building on, you know, a bit more of a unified rating scale, so we get alignment across industry to actually be able to compare some apples to apples, whether it's genetic resistance or just surveying, so that way we can actually see, okay, this field is this bad, this field is this bad, what are the differences for that? So that's a big success. Conversely, what's our biggest opportunities and challenges is, I think it's got to go back to verticillium again in terms of what are the major threats for at least Manitoba canola production, and potential threats in Saskatchewan, maybe ultimately Alberta if it travels that far. I don't know, luckily I think we're treated by some more westerly winds that keeps it maybe in Manitoba, but in terms of, yeah, what's going to be a threat that's going to lop off our upper yield potential, I think that's one of the key pathogens we got to focus on.
I was just adding on to that, it would be nice to see some association or some benchmarks set to when we see this level of disease, this is the expected yield loss, and maybe that takes, I mean, when you say you see a devastator, you see everybody's seen it in the field, but we don't have a good idea of what that cost us, I think. And so that might put some minds at ease, or it might not, but at least we would know. So that would be nice to see.
Yeah, and the challenging thing too, and Evan touched on that, is that verticillium and blackleg often coexist together in the stem. And so trying to separate the yield impact from one versus the other is difficult. And recently, Saskatchewan, through a sponsored testing program by Sask Oilseeds, showed that pretty much all verticillium samples that were sent for testing did have blackleg in them. And so trying to test that, and also, you know, for breeding programs or yield evaluations for registered cultivars, it gets pretty murky. Yeah.
So I'll ask one more question, then go to audience questions here. Just based on the 2024 season, what's kind of your biggest takeaway as we head here into 2025? What's top of mind as we head into next year?
I don't want to go first. Okay, so one takeaway from 2024 is it kind of made me take a look at that classic disease triangle a little differently this time. And you know, we need the susceptible host, we need the environment, we need the pathogen, right? But also it's the timing of all these things happening together. And so we took a look, you know, early in the season. We had a very wet spring in terms most of Manitoba. And so that was excellent conditions for some early season blackleg. And then coming into late June, we saw all those rains also happen, and all of a sudden, great, that's excellent conditions for sclerotinia. We got hammered there. And then all of a sudden we kind of tapered off with, you know, fairly dry conditions and some hot weather, which is great for late season development of verticillium. So looking at all those kinds of conditions and the timing of those really kind of put into perspective that we got to evaluate our risks as time goes on and those combinations kind of interact together.
Yeah, I think you said it much better than I would have, but yeah, Mother Nature kind of dictates how things are going to go, and this year was just, yeah, everything kind of aligned for all of these to stack on top of each other. So in a contract research position, I'm very concerned about getting the perfect timing, perfect applications. Everything needs to be just right, and sometimes Mother Nature is just going to do what she's going to do anyway. So, yeah.
I think you guys mostly covered the thoughts on disease from my side. The one thing, I just had a conversation with someone the other day on our move to more pod shatter resistant technology in most products. How much is that actually, I don't have a number for this, but how much is that actually protecting yield loss from these diseases? So in older varieties, when you had a lot of disease, would those just be shattered pods and gone when they mature early? So is that really kind of keeping us at a level that's a little bit better than it might even be if we hadn't made that move? I'm not sure, but just some food for thought.
Okay, so I think we've covered a lot. We've done our 3 of 3. I'm going to open up here for audience and online questions, if there are any, for our panel. So yeah, feel free to raise a hand if you have some questions.
Hello, yes. While we wait for, I don't know, I saw Jeanette's hand go up, so I'm going to interrupt her first. Are we encouraging more verticillium stripe infection because we are doing more straight cut, meaning harvesting later, leaving the crop out longer than if we were to swath first?
Some people say yes, some people say maybe, I think. You know, we've got to take a look at some of the evidence and the data, and right now that data does not exist in a reliable form. I know there's projects that are going to be probably coming down the pipeline to actually assess that. Whether it's, you know, say the genetic background, or maybe more likely, you know, that disease can progress in standing canola that helps return that inoculum back to the soil. A lot of unknowns there, but it's something that's definitely of interest and is also one of the key research priorities for the Verticillium Steering Committee, which helps direct research funding to some capacity.
I was just going to ask, and I think this isn't just Manitoba, when the provincial survey reports came out, the sclerotinia prevalence seems to be quite low compared to what agronomists or farmers are saying what they felt like it should be. Any thoughts on that?
Yes, that's a, I guess, me question. I think a big part of the survey too is in terms of timing. Did we necessarily catch sclerotinia like to visibly see it versus when some of the other infections might have come in? As well too, we're doing, you know, we're picking a field out of an RM, and it might be, we're doing, let's say in one RM, 3 fields, and it might not be one that had sclerotinia. So I think those are some of the things that might have been a lot more prevalent than maybe the disease survey captured this year for sure, as we, you know, saw it just driving around the fields.
One other thought on the disease survey side of things, not to pick on it, but you mentioned the window of timing. I really have to question with what we talked about with verticillium, how late it can infect. Are those numbers actually lower than what you would like if we would really see if you went right at harvest to every field? Whether that's actually causing a measurable yield loss or not, but I think your incidence of verticillium, I feel like it's really close to like 100% in Manitoba, but we don't see that on the survey because we're not there right at the end.
Thank you all for your insights. My colleagues and I just thought we noticed on our farm that there might have been a correlation of higher amounts of verticillium and or blackleg in fields that we had to protect against flea beetles. So just wondering if anybody else saw that sort of correlation. We were wondering if that flea beetle bite mark allowed an opening for those diseases to infect.
So originally, I think there was some research done some years ago, took a look at, you know, is wounding essentially a key entry point rather than say the stomata or getting into the cotyledons with blackleg. And recently there was actually a paper published by Delantha Fernando and I think Gary Peng from AFC to reinvestigate this. And essentially, I mean, all this research is also very dependent on the site years and the conditions that are provided in their testing phases, but not significant impacts, really. Probably there's other factors at play, like say probably amount of inoculum that have a much larger role for at least blackleg. I can't really say for verticillium, considering it's a soil-borne pathogen, it's actually coming through the roots, so I don't really see any reaction directly between that, but it also could be, you know, based off an overall like plant stress responses. You know, if it's trying to recuperate coming from severe flea beetle feeding, maybe it's more vulnerable towards other stressors like biotic stresses and verticillium. That's just my thoughts.
Hello, I'm Vikram Bisht. I work with potatoes. And in the potato system, we see that the Verticillium delia infects the roots very early in the season. The symptom expression depends on how hot or dry the conditions are. I expect something similar happening in canola as well. When you go after the harvest, almost every plant appears to be infected, but if you go when the plants are still nice and green, you don't see much infection. So it's not the infection that is taking place later, it's the expression that is showing up.
So apparently no question.
I-
Anybody with questions?
That is one interesting point of hot and dry, and it's been talked about that verticillium is higher in hot and dry years, which is, mentally a bit backwards from what you expect from most diseases. So.
Thanks so much for the discussion. I was just wondering, so there was a lot of talk on verticillium, blackleg, but I think blackleg was once a minor disease just like clubroot, and based on the complexity of Plasmodiaphora, it is a Plasmodiaphoromycetes, it's not a fungus. Right now I don't think there's effective fungicide, and I don't think 2 years crop rotation can manage the disease. I'm not sure about the evidence, because most of the time the resting spores can survive up to 10 to 20 years. So, and in the wake of climate change, since the temperature is getting warmer in the temperate regions of the world, just like in Canada, what do you expect in next 5 years about clubroot, and what are the initiatives being taken, either in terms of breeding or agronomic practices? Yeah.
So you kind of jumped around some pathogens there. We're talking about clubroot and spores in the soil?
Yep.
Okay. After 2 years, the amount of spores in the soil decreased by 90%. And that is based off of some pretty sound research coming out of Alberta. And so when we're taking a look at, say, a 2-year gap for that, that does actually decrease severity quite enough that you can actually reduce that. So that works, but I wonder if anyone else on the panel wants to follow up on the other questions.
I think, yeah, as long as your initial spore load isn't billions of spores, then reducing that by 90% is still going to get you below that infection level. But yeah, continuing to monitor and crop rotation is what's going to prolong our the current resistance genes that we have. If we just think that the breeders are going to come up with something new next year when when something breaks down, like, we do have to be caretakers, I guess, of the tools that are given to us.
Yeah, on that point, there is a pipeline of genes that are not necessarily deployed yet in Western Canada, so there is, I guess, some backup there that can come to market in the next little while, and there are different companies are working with different genes now. It's not the same level as blackleg, where that's maybe published in some cases, where you can knowingly go to a different gene or something like that. Right now, most companies are just, and it's more of a marketing gene 1, gene 2, whatever it's called. So that would be another step that could be taken some point in the future that would potentially help with like knowing what's out there, but that the clubroot side isn't at that level yet.
It probably will get there eventually, I think, because when we're talking about first gene, clubroot resistance, it's a single gene from Mendel. Second generation, it's actually different genes depending on which company, right? So it's much more complicated for that. The challenge with clubroot is that in a clubroot gall, you may have different races present, so you need to work on single spore isolates to identify the dominant race, and that is very tedious work. And then you have to distribute that single spore isolate to all the breeders, so they're working with the same material and advancing that. So that's a key research focus that the clubroot steering committee is helping coordinate for that.
So the question here is there any way that we as retail agronomists can contribute to the verticillium research?
Okay, I'm jumping in here again. I would say definitely reach out. If you have farmers who are willing to participate in our disease survey, we also have some verticillium specific surveys that we're hoping to implement for 2025. I'd highly recommend reaching out. I would love, because we're just trying to really start the tracking and to start putting together the severity rating, have an idea of how it's affecting here in Manitoba. So I would say that would be a first step for 2025. That would be great if you got in contact with me. Anyone else?
I hate stealing the mic a bit, but I really want to give a big shout out to Manitoba Canola Growers Association, who has an on-farm research trial program, along with a lot of the other commodity commissions in Manitoba. And next year, they're looking to introduce a verticillium trial. So if there's any interest from retail's agronomists that want to participate and have growers interested in this, that'd be an excellent place to get on-farm results to investigate verticillium potential management techniques, like we're talking straight cut versus swath.
So I think with that, we're going to end the panel here. I think we'll be around for a couple more minutes if there's any other questions. But I want to say a big thank you to the panelists, and I'll turn it over to Kim.